Hello friends, welcome to SETI Botany classes. This is video part 2 of general characters of Pteridophyta. In part 1, we have already discussed habitat, morphology and formation of sporangium in Pteridophyta. In this video, we will discuss formation, structure and types of gametophyte, sex, organs, Antheridium and Archegonium, and then what is the structure of Antheridium and Archegonium, structure of antherozoids, how fertilization occurs, and what are alternation of generations. Now, this we have already discussed in the last video part 1 of general characters of Pteridophyta, that sporangia, they are born on the leaves, and leaves bearing sporangia, they are called as sporophylls. And if we see the structure of each sporangium, this is bounded by a jacket called as sterile layer, and this is the tapetum layer, which is present inner to the wall layer, and this is helping in nourishment of the spore mother cell. The spore mother cell undergo meiosis to form haploid spores. Sporogenous tissue may give rise to spores. All are of the same size, shape, and morphology. Structure is also same. Then it is called as homosporous condition, like in Xylotum, Lycopodium, Equisetum, and Teres. Now, spores may be different in shape, size, and structure, like in Selaginella and Marsilia. Smaller spores, they are called as microspores, and bigger spores, they are called as megaspores. Microspores are produced inside microsporangium, and megaspores, they are produced inside megasporangium. Now, this is the sporangium of Teres, where homosporous condition is found. This is the spore of the Teres. All the spores, they are of the same shape, size, and structure, and this will germinate form gametophyte. Bisexual gametophyte will be produced if homosporous condition is there. And this is in case of Selaginella. There are two kinds of spores. Smaller one are called as microspores, and bigger one they are called as a megaspore. So production of two kinds of spores is also called as heterospory. So in case of Selaginella, microspore will germinate to form the male gametophyte, and megaspore will germinate to form the female gametophyte. But if condition is homosporous, spore will germinate to form the bisexual gametophyte, and if condition is heterosporous, spores will germinate to form unisexual gametophyte. Now, germination of the spore may occur inside the sporangium only, and if they start germinating inside, this is called as endoscopic, and if it comes out and germinate on the soil, this is called as exoscopic. Now, spores after dehiscence of the sporangium will fall on the soil. When they will get suitable temperature, moisture, and nourishment, they will start germinating. Now, this is the bisexual gametophyte of Teres. This is bearing both the sex organ, but they are separated by space. Here in the apical part, generative region is there, and this bear female sex organ called as Archegonium, and this is the posterior part of the gametophyte of the Teres. This bear the sex organ called as Antheridium. Now, this is called as prothalis of Teres. Prothalis is also called as gametophyte, and this is being produced after germination of the spore, and prothalis or gametophyte is attached to substratum by means of rhizoids. Rhizoids, they help in fixation of the prothalis or gametophyte to the soil and absorb water and minerals for the plant. And this prothalis is also heart-shaped and haploid in genetic constitution. Now, this is the Antheridium of the Teres, having sterile jacket, androcyte mother cell. Now, androcyte mother cell, they will form multiflagellate antherozoid. Because of the flagella, this can move from one place to the another place. Now, this is germination of the megaspore. Megaspore is showing endoscopic formation of the gametophyte because germination starts inside the megaspore. So this is also called as endoscopic gametophyte. This is in case of Selaginella, and this also occurs in Marsilia. This is the example of Selaginella. And here see only one kind of sex organ is found, that is female sex organ, Archegonia. So plants which show heterospory, microspore will produce male gametophyte and megaspore will produce female gametophyte. So this is the megaspore, which is germinating to form the female gametophyte because germination is occurring inside the megasporangium. This is called as endoscopic development. But when the spores they fall on the substratum and start germinating, this is called as exoscopic formation of the gametophyte. This happens in case of Xylotum, Lycopodium, Pteridium, and Teres. In both the cases, because spores they are being produced after meiosis. When spores they germinate, whether they are germinating inside megaspore, microspore, or they are falling on the substratum and germinating, they will be haploid in genetic constitution. Now, this is the exoscopic gametophyte of Pteris, or we also call it as prothallus, heart shape. We have already discussed this is the female sex organ, and this is the male sex organ attached to substratum by means of rhizoid, and this is green in color. Because it is green in color, so this is autotrophic in mode of nourishment. This can make its own food, and rhizoids will help in absorption of water and minerals for the plant. And some of the prothalli, they may not be green in color in some of the genera of the Pteridophyta. And when they are not green in color, they will not be they will not be able to make their own food. So when they are not able to make their own food, they are also called as heterotrophic in mode of nourishment. So they may be saprophytic, that is, they may be getting their food from the dead organic matter. An example is Xylotum and Lycopodium. Now, this is the saprophytic gametophyte of Lycopodium. Here we will see that this prothallus, when we see morphologically, this appears like this, and this is non-green in color, saprophytic, attached to substratum by means of rhizoid. If we see its internal structure, you can see these are the sex organ born on the prothallus or gametophyte. So this is again bisexual. This is the Antheridia. These are Antheridia, and these are the Archegonia. And this part of the prothallus or gametophyte is showing association with the fungus. So this association with fungus is called as a mycorrhizal association. Fungus will act like a sponge because it is saprophytic. So this will be attached to substratum by means of rhizoid, and this will absorb nourishment from the dead organic matter with the help of fungus as well as with the help of rhizoids. Fungus will act like a sponge, and this will absorb the nourishment and give to the prothallus, and ultimately sex organs will develop. And this is the male sex organ Antheridium, and this is the female sex organ Archegonium. If we see the structure of the Antheridium, this is bounded by a sterile jacket, and inside it has androcyte mother cell, and androcyte mother cell will metamorphose to form antherozoid. So this is the antherozoid, which is bearing large number of flagella. This is elongated nucleus, and remains of cytoplasms are also attached to the antherozoid. Now, this is the female sex organ called as Archegonium. Archegonium, if we see the structure, this is flask shape. This lower part is venter, and this part is called as neck. So in the venter is present egg cell. This is venter canal cell, and this is binucleated neck canal cell. And this is the jacket which is surrounding the Archegonium, and this is attached to the prothallus by means of cells. At maturity, all the cells, that is venter canal cell and neck canal cell, will degenerate, and mucilage will be produced, and this mucilage will attract enterozoid which are multiflagellated toward itself, and this will travel through the neck of the Archegonium and reach toward the egg cell. So ultimately, when all the cells they degenerate, enterozoid will travel toward the egg, and fusion of egg and enterozoid nucleus will take place, and diploid structure zygote is produced after fertilization. Because fusion has occurred, syngamy has taken place, so egg and enterozoid have fused together. So zygote will be diploid in genetic constitution. Now, zygote is the first cell of the sporophytic generation. Now, this will undergo mitotic cell division to form the sporophyte. So this is the zygote. Different type of development of sporophyte occurs at different species. Now, this is the zygote. This will divide to form two cells. This is the upper cell, and this is the lower cell. Now, upper cell is going to form the suspensor. Now, lower cell further divides into two cells, then into four, then into eight. And this part, lower part, will form the proembryo part, and this upper part is forming the suspensor part. The proembryo will further undergo division, like first two cell, 4, 8, 16, 32, 64, and so on. This will form multicellular structure, and this multicellular structure start differentiating. This part forms foot, which will provide nourishment to the embryo, and this part start developing into stem, and this part is where root will be produced from the proembryonic part. So this whole development is taking place inside the Archegonium. So this is second type of development, like this is dividing to two cells, 4, 8, 16, 32, and so on, and ultimately this will develop into young sporophyte. And here also, this zygote is undergoing multiple divisions, differentiation occur. This part is forming stem, foot, root, leaf, differentiation occur, and this is the sporophyte. This is the sporophyte. So we can say development of embryo, or you can say sporophyte, is occurring inside the dilated part, that is venter of the archegonium, and this is occurring inside. So this is called as endoscopic. And if the axis of the embryo is directed inwards away from the archegonial neck, like in case of Lycopodium and Selaginella, right? This is called as endoscopic. Now here, this first part is endoscopic. Now, if the axis is towards the archegonial neck, like if axis is toward the archegonial neck, this is called as exoscopic ecosystem. Like development of stem is, if it is on the lower side, this is endoscopic, and if it is on the upper side, like here it is on the upper side, this is exoscopic, and if it is attached at right angle, then it is called as lateral. So we can say formation of sporophyte, you can say development of embryo may be endoscopic, this may be exoscopic, this may be lateral. So ultimately, sporophyte is produced from the zygote. So this will be deployed in genetic constitution. Differentiation of the tissue occur. Some of the cells, they may differentiate to form stem. Some of the cells will form root, and some of the cells they form foot, which is basically helping in the nourishment. So ultimately, all the tissues will get differentiated from this embryonic part of the sporophyte, and young sporophyte will develop into mature sporophyte. So embryo, as we have discussed, will undergo mitotic cell division and differentiate to form a young sporophyte with differentiated tissue. Some of the tissues, they may form stem, leaf, root, and some of the tissues, they help in nourishment of the young sporophyte. But later on, a young sporophyte will develop leaves, and this will turn autotrophic in mode of nourishment. Root part will develop into root. This will help in fixation of the sporophyte to the substratum, and this will develop into mature sporophyte. So in the whole life cycle, we have seen that main plant body of the Pteridophyta is sporophyte. Now this will reproduce by the formation of the sporangia. Sporangia can be of same type, they can be of different type. Leaves bearing sporangia are called as sporophylls. The sporangia will produce spores. Spores may be homosporous condition. It can be heterosporous condition. When homosporous, this will produce one kind of spore. When heterosporous, this will produce two kinds of spores. Spores will germinate to form the gametophyte. Now, gametophyte will produce sex organs. If spores are of same type, they will germinate to form bisexual gametophyte. This will bear both the sex organ, Antheridium and Archegonium. Antheridium is the male sex organ. Archegonium is the female sex organ. Antheridium will produce entherozoid. Archegonium will have haploid egg. And if heterosporic condition is there, microspore will germinate to form male gametophyte. Megaspore will bear female gametophyte. Ultimately, entherozoid will travel toward the egg. Fertilization take place. Zygote is produced. Zygote undergoes cell division, mitotic cell division to form sporophyte, young sporophyte, and mature sporophyte. So sporophyte has given rise to gametophyte, and gametophyte has given rise to sporophyte. This is called as alternation of generations. Now, because prothallus structure or gametophytic structure is different from the sporophyte, morphology of sporophyte is different, morphology of gametophyte is different. So this is called as heteromorphic type of alternation of generations. If you like my video, please like, share, and subscribe. Thank you for watching my video.